So I do this. Oh, okay. Then I go to my email.
Yeah, remember it's recording. Then you can just minimize that and go to Zoom. Yeah. You can just minimize it. In case needed.
About 5 more minutes and then we'll get started. Okay, why don't I go ahead and get started with some of the preliminary comments. For those of you who have not yet received your graded second exam, be patient. I expect to have them all finished by Thursday and sent out to everyone. Part of it was a logistical issue. Part of it's also that I'm writing more informed comments on your exams when you have missed questions. so you can understand exactly why you missed questions on a particular exam. Trying something new this time. I'm not going directly through WVU's portal. Instead, I'm using a sort of a combination of WVU as well as the internet. The free version of Zoom is supposedly limited to 40 minutes, but I'm hoping that by using the internet, but going through WVU's system, we'll be able to have unlimited time. We'll see what happens. Again, I'm recording these presentations, which the number one priority after I finish this lecture is to finish grading the exams. And then the second priority is going to be figuring out how to upload these to YouTube. When I've got it uploaded to YouTube, I will send out a message to the entire class telling them how they can access them. If those of you that are listening, if you can suffer through hearing me speak a second time, more power to you. I trust that everyone is staying healthy and is practicing distances between individuals. Hopefully that will remain. I have not heard a word yet regarding what's going to happen in the fall, whether or not fall classes are going to be held or not. I suspect they may be held, but if so, they may be in a modified version. Today, what we're going to do is to finish up the discussion on forensic biology. I have taken out some slides that really I think weren't essential for the meaning that I'm trying to get across in these lectures for both the forensic biology, and then we're going to move into understanding horizontal gene transfer. And I've also edited that compared to what your PDFs are, so we can focus on the main points, or at least what I think are the main points. We left off with a photograph of Thomas Jefferson, and what we're going to talk about today is the evidence that Thomas Jefferson sired children with one of his slaves, Sally Hemings. Thomas Jefferson had no acknowledged male heirs, meaning we couldn't find an acknowledged Y chromosome from him. He did have a paternal uncle, however, who did have male heirs. So they were able to obtain DNA samples from these so-called cousins, distant cousins. The Y chromosome should be the same as what Jefferson has. And that's what they used to examine some of the offspring, some of the individuals that thought they were descended from Thomas Jefferson. The family that made the most noise about being descended from Thomas Jefferson was Thomas Woodson, a child born on the Jefferson plantation many generations ago, and it's the offspring of this family. When they did a comparison of several descendants from Thomas Woodson with the Jefferson Y chromosome, there were no matches, indicating that Thomas Woodson's family was not descended from Thomas Jefferson. When they looked at some other individuals descended from Easton Hemings, a son of Sally Hemings, they did indeed find several descendants that had the Y chromosome, similar to what Thomas Jefferson had. And one male line descendant. Now, the press has, in my opinion, overstated the strength of the evidence. They state that the DNA proves that Thomas Jefferson was the ancestor of some of these descendants. I think they're overstating the value and the strength of the DNA evidence. The one thing we can conclude without question is that Thomas Woodson's offspring were not descended from a Jefferson. Easton Hemings' descendants, at least some of them, are derived from someone carrying the Jefferson Y chromosome. There were 25 potential Jefferson donors, if you will, living in Virginia at that time. That is, there were 25 individuals carrying the Jefferson Y chromosome, any one of which could have been the father to Sally's children. Most historians, however, and I concur, think that the circumstantial evidence as well as the Y chromosome evidence strongly indicates that Thomas Jefferson probably was the ancestor of some of Sally's children. However, the DNA alone would never hold up in court because there were several other individuals that could have sired the children. But again, taking the circumstantial evidence into consideration, it makes a strong case that Jefferson was indeed the father. And on popular TV shows and things like that, the focus is almost entirely on the DNA, which I think is unfortunate. I think presenting more of the circumstantial evidence would present a more complete picture and strengthen the case that Thomas Jefferson was, in fact, having sex with his slave. By the way, if you didn't know, Sally Hemings was half-sister to Thomas Jefferson's wife. They shared the same father. Sally's mother was a slave owned by the man that fathered Jefferson's wife. He also fathered Sally. So they had a striking resemblance to each other. Okay, DNA is not just used with humans, identification of humans, but it can also be used in other situations in forensic science investigations. Plants, for example. There was a homicide in 1992 with a suspect, Mark Bogan, who claimed he had never been to the property where the homicide was committed, but in his truck, there were seed pods from a tree, Palo Verde, that was found in his truck. These seeds actually clearly came from a tree at the scene of the crime. Therefore, his claim that he'd never been at the scene was a lie. So it was identical to the seed pods in the suspect's truck. It linked him to the scene of the crime, and he was convicted. Snowball. Those of you, certainly the forensic biology students, I'm sure have probably heard about Snowball. Snowball is the name of a cat. 1994 in Canada, Prince Edward Islands in Canada. There was another homicide. Shirley Dugal, I think, the strange wife of Douglas Beamish. How's that for an English name? She was murdered. They found hairs from a white cat on Shirley, the victim. Douglas owned a white cat named Snowball. Tests showed that they tested Snowball as well as 20 other cats to look at STR analysis. It was clear that the cat hairs, came from Snowball. Today, there are 11 STR loci that are used in identification of cats. Remember, transfer of evidence. Presumably, Douglas had the cat's hairs on his clothing. When he committed the homicide, he transferred some of those hairs onto Shirley's clothing or body, and that's what the police found, and they were able to make a linkage between the cat and Shirley, Douglas's cat, as well as Shirley. As I say, today there are at least 11 STRs that are used in cat identification. In the US, it's estimated that there's about 65 million cats. A similar set of studies have been done on dogs with STR analysis also. So both dogs and cats, if you transfer pet hairs during the commission of a crime, there's a good chance they're going to be able to trace it back to your pet. Homicides. Mostly what we've talked about is the use of DNA evidence in homicides, rape homicides, or rapes by themselves. But usually it's a very serious crime. There was a British study where they found that 75% of individuals breaking in to homes, burglary, left their fingerprints. When shows like CSI and whatnot came on the air, there was some public concern that what we were doing was to be educating criminals how to break the law and not get caught. Well, we've been using fingerprints in this country for well over 100 years. And 75% of the geniuses that are committing break-ins don't have enough common sense to put on a pair of gloves. Instead, they leave their fingerprints behind. Additionally, as I say, so much for CSI educating criminals. Criminals tend not to be the smartest in the population. There are several stories about individuals that, rob a liquor store and they're wearing their softball jersey with their name on the back. There's a story of an individual who robbed the cash register, and the quick thinking clerk said it was a liquor store. The guy wanted some bottles of liquor. And he said, yeah, but I got to make sure you're 21 years old. And so the idiot gave him his ID, for which they got the name. Here in Morgantown, a former football player broke into an apartment along with a colleague and stole computers and things like that from the inhabitants of the apartment. Only problem is the idiot wore his practice jerseys from the football team, which had his number on it. So they were able to quickly identify him. He got arrested and convicted. Criminals are not the smartest people in the book. Not only do they leave fingerprints, but about 50% of them leave some kind of biological sample containing DNA evidence. I don't fully understand this, but apparently frequently during the commission of a break-in, the perpetrators feel compelled to eat the food they find in the refrigerator, and oftentimes they will urinate and defecate all over the place. And of course, DNA evidence can come from all of those places. So clearly, STR analysis can be used not only in major crimes such as rape and homicide, but in relatively minor ones to provide evidence to identify perpetrators. In Britain, they keep fairly extensive DNA records. A colleague once told me that if you were arrested in the UK for spitting on the sidewalk, they're going to take your DNA and put it in their CODIS system, their system for a database of STRs. Turns out, at least in Britain, crime in fact runs in families. The STR profile you obtain may not match one that's already in the database, but odds are pretty good that they have a close relative. And so that can be used to track down and identify the true perpetrator. And DNA of relatives. I'm sure you've heard there have been several convictions or several clues that led to identification of a perpetrator using relatives that have done 23andMe and these various other studies on people's ancestry. They end up in a database, and that database is often shared with crime scene investigators. And there have been several convictions, several identifications of a close relative that then led to the identification of the perpetrator, and they have been convicted. Some of the problems with PCR analysis. One of the biggest ones is cross-contamination, especially if you have a very high-profile case. It seems that every cop in the vicinity wants to show up and get his face on TV cameras when that happens. And that significantly increases the likelihood of cross-contamination. That is, individuals entering the crime scene after it should have been secured and leaving their own biological evidence behind. Yeah, the crime scene. Could happen in the lab where sloppy work on the part of the lab technician results in his contaminating the samples. That's usually relatively easy to control for because most labs require DNA examiners and other people that handle the evidence to surrender their DNA for STR profile. And so that's relatively easy to identify, and typically it's only one individual that's causing the contamination. One of the other issues is the quality and quantity of the DNA samples. Unlike a lot of shows like CSI, again, crimes are not committed in the most pristine environments. They are often subjected to environmental insults to the DNA, causing degradation. So oftentimes the quality is not what one would desire. The reporting of statistics can also be a problem. There are several different approaches. We're not going to talk about it, but at least the last time I looked, there is no unifying way to report statistics. CSI effect is a real thing. Jurors, lawyers, judges, they all expect lots of forensic evidence, and in particular DNA evidence. And this has resulted in crime scene investigators collecting anything that might be of probative value, that is, that might contribute to the solving of a case. And because of that, sample collection has gone up significantly, resulting in a significant backlog in terms of processing samples. Rape cases in particular, there's a huge backlog, especially in New York City. One of the major caveats, however, with STR analysis is that DNA does not provide any temporal linkage. It doesn't tell you when the sample was deposited at the crime scene. It could have occurred months before the commission of the crime, could have occurred a week before the commission of the crime. All we know is that the person had visited the place where the crime was committed sometime in the past prior to collection of evidence. So it provides, STR analysis provides for spatial linkage, that is in space, having been in the site of the crime scene, but it doesn't provide any temporal linkage. Privacy issues. The original 13 STR were selected for being non-coding sequences of known function, and thus it was felt that no medical information could be derived from it. Now, there is a small caveat. There may be correlation. You may see a particular STR allele is strongly correlated with a particular disease. As the STR loci have been expanded, there are a couple of them that are actually found inside genes and could conceivably provide some information regarding an individual's health. There are private DNA databases that we just talked about, National Geographic's, 23andMe, various things like that. Generally speaking, most of them appear to have no qualms about sharing their database with investigating individuals investigating the crime. There's also databases of ethnic ancestry, indicating what the ethnicity is of the individual. There's a fairly famous story about an individual that appeared to have been the greatest criminal to ever live. They kept finding this woman's STR profile in a series of murders, burglaries, robberies, all kinds of places, including rapes. Mostly in Europe, Austria, France, and Germany. Way too many cases for a single individual to have committed, even a very dedicated criminal. Turns out that the swabs used to collect the samples were contaminated with this woman's DNA. She worked in a factory in Austria that produced the swabs that were used. And sloppy behavior resulted in her DNA contaminating a lot of the swabs that were used for collection of evidence. Okay, if you are in the field of forensic science, hopefully you've already seen this YouTube presentation. Maybe it's not YouTube. It's by John Oliver. If you don't know John Oliver, he broadcasts on HBO. He does a superb job. He blends comedy with social commentary. And this is a fantastic example of it. It's about 19 minutes long, so we're not going to show it now. But I strongly encourage everyone, even if you're not a forensic science major, to listen to this presentation by John Oliver. Based on my experience with 20 years being involved in the field of forensic science, it is absolutely dead on in terms of its criticism, in particular hair analysis, some of the other analyses that go on. Sort of, I've always said that the field of forensic science is a bit of a misnomer. The forensic investigation can be divided into two subcategories: forensic science, which is primarily forensic chemistry and forensic biology, and forensic opinion, where the evidence is evaluated by a subject expert, where there is virtually no scientific evidence to support the conclusions. Typically, individuals doing that, mostly impression evidence like fingerprints, tool marks, bite marks, hair analysis, various things like that. The expert witness, which is determined by the court in each case, who qualifies as an expert witness. You will often hear the witness say, to a reasonable degree of scientific certainty, which is to say, I don't know, I'm making a guess. But this is an excellent presentation. It's entertaining and it's very informative. And as far as I'm concerned, absolutely dead on. So what's on the horizon, or what is actually being utilized right now? The STR analysis works best if the suspect is in the DNA database. As I said, STR analysis sometimes can give you some indication of the ethnicity of the individual. If somebody is not in the database, you hope that they commit another crime and the DNA gets entered into the database, or a relatively close relative does one of the National Geographics or 23andMe, and the investigators happen to search that database and find a close relative. Another way of approaching this is to look for SNPs, single nucleotide polymorphisms, where you don't have an eyewitness to what's going on. But the SNPs can tell you lots of information, such as the natural hair color of the individual, the extent of pigmentation, eye color, various other things that are encoded by the genes. But most importantly, pigmentation ethnicity is the ethnicity, identification of the ethnicity. If you have an individual that has blown themselves up in a suicide bomb, frequently there is virtually nothing left of the perpetrator that could be used for identification purposes, but SNPs can be used to identify the ethnicity. If it's a Muslim-related suicide bombing, there are several countries that the perpetrator could have come from, but SNP analysis allows you to determine whether they are of Eastern European origin, the Middle East, or various other countries. This allows for exclusion of a lot of other potential suspects and focusing only on those that came from the ethnic group that the bomber came from. It also provides an unbiased molecular witness. Once upon a time, I proposed doing this. The deans at WVU decided that it was too touchy in terms of the social ramifications of identifying people and classifying them based on ethnicity, so we didn't pursue it. As I said, this can act at least as an exclusionary test, which is very valuable in excluding populations that you know the perpetrator did not come from. So you're not wasting your time chasing leads that are not going to prove anything. Yeah, exclude certain groups. Okay, that finishes what we're going to talk about in terms of forensic biology. And now we are going to turn to horizontal gene transfer. What we've been talking about thus far is almost exclusively vertical gene transfer. That is, transfer of genetic information from parents directly to offspring. Horizontal gene transfer refers to acquiring DNA sequences horizontally, typically from a different species. There is an organism that's related to malaria. Whoa, this is not the beginning of the lecture. Sorry. Let me get out of this and go back to the beginning. I was making modifications, and unfortunately, I did not go back to the beginning. My usual caveat that the PowerPoints are essentially an outline. They are not intended to replace participating and listening to the lectures and or using a textbook or online references. Horizontal gene transfer. Genes or DNA enter another genome and become part of that genome. This is the primary reason that antibiotic resistance is such a problem, because gene transfer, horizontal gene transfer, is a very common phenomenon in unicellular organisms. Once antibiotic resistance develops within a bacterium, it will quickly spread those genes for resistance to other members of its species, as well as other species of bacteria, whether it be plasmids or phage spreading it. It's highly significant in single-celled organisms, not just prokaryotic organisms, but fungi, yeast, things like that. Single-celled organisms apparently swap DNA fairly frequently. Genes from chloroplast and mitochondria often migrate to the nucleus. This led to an endosymbiotic theory of how chloroplasts and mitochondria came into being. It was thought that these organelles were originally bacterial endosymbiotes. That is, they had a symbiotic relationship with the ancestral eukaryotic cells. Ultimately, it went from essentially a symbiotic relationship to an obligate relationship between these organelles and the nucleus of the eukaryotic organism. One example, the malarial pathogen at some point in time acquired DNA from humans that facilitated its ability to stay on in the human body. Prior to the acquisition of that gene, it got purged fairly quickly. Nuclear DNA and mitochondrial DNA originally originated as different organisms. That's basically the basis of the theory. Mitochondria are thought to have been derived from proteobacteria, whereas the chloroplast are thought to have been derived from cyanobacteria. And it happened a long time ago in history. In the case of chloroplast, 1.5 billion years ago. Gene transfer. In the mitochondria, there are roughly 1,515 proteins in the mitochondria itself, a functional mitochondria, yet only 37 genes, 13 of which are proteins, are actually encoded in the mitochondrial DNA. The rest of the proteins are derived from genes that were transferred from the original symbiotic bacteria into the host nucleus. One other interesting observation, we talk about the genetic code being universal. Well, here's an example where it is not universal. UGA, genes encoded by the nucleus, encodes for a stop codon. It will terminate translation. In the mitochondria, however, it encodes for a tryptophan. So it has taken a stop codon and it is now a sense codon, a nonsense to a sense codon. There are only 22 tRNAs in the mitochondrial DNA. That's true of both humans and yeast. There's a lot of conservation. There are other differences as well, but many of these are species specific. Some are conserved, many are species specific. So the question arises, why retain any genes? Well, part of it is there's a difference in code as well as processing of the messenger RNA, so that would have to be changed for a gene to migrate to the nucleus. But perhaps the biggest issue is that hydrophobic proteins are difficult to move through the cytoplasm, the aqueous cytoplasm. Redox reactions repairs anything that goes wrong. are very time consuming if they needed to communicate with the nucleus, get transcription, translation, and then movement into the nucleus itself. By having some of the genes present, that time is diminished. There are a few examples where mitochondria have entirely lost their DNA, but that's pretty rare. There's only a few examples that I'm aware of. There's evidence that plastids, the precursors to chloroplasts, were only captured once in the evolution of plants. Evidence suggests that mitochondria, however, were captured multiple different times during evolution. When I was your all's age, I used to want to be a science fiction writer. And so I would come up with all these ideas of, you know, what would be required, what would make a compelling story, what would project science, a reasonable projection of science that was on the horizon. And I used to think if we're going to colonize other worlds, it would be awfully nice if we could figure out a way to do photosynthesis so we don't have to worry about planting large gardens and things like that, because the soil may not be very useful in terms of, at least initially, in terms of yielding crop and whatnot. There have been several science fiction movies out in the last decade that featured this. One in particular used potato farming to get around the lack of crops. So I thought one of the requirements would be we'd have to figure out how to do photosynthesis. I thought the other one would be to tailor our gut genome to make modifications to fully exploit the new environment. And I still think that those two are two criteria. They're two areas that are going to have to be addressed before we're going to successfully colonize some other worlds at least. So that was my science fiction. So turns out that there are several animals that can actually feed themselves through photosynthesis. And one example is a sea slug. The sea slug takes the plastids, which are the precursors to the chloroplast, from algae. Algae is a eukaryotic organism that has chloroplast and can photosynthesize. And the sea slugs actually steal them from the algae. They become sequestered in the sea slug's digestive epithelium, where they're exposed to sunlight. They have captured, the sea slug have actually captured some genes from the algae's nucleus, genes that are required for long-term survival of the plastids, so they're not turning over very rapidly, that can survive for several months. Allow the plastids to photosynthesize for months on end. So the sea slugs are animals that don't have to feed for several months at a time because they can make their own food through photosynthesis. So the thing I fantasized about, if you will, when I was in high school and college has shown to be a reality. And who knows, maybe someday we will start doing something similar to that. Be sort of nice if you were hungry just to be able to go out and stand in the sun and make your own nutrients that's going to lead to ATP production. Of course, we have to deal with skin cancer, how to treat that, but I think that's possible too. Here is the sea slug. You can see it's feeding on the algae in the upper left photograph. And on the right photograph, you can see that the epithelial cells have become populated by the plastids, those green dots throughout there that's going to allow for photosynthesis. I've included a YouTube video that you can watch that shows the sea slug sucking the plastids out of the algae. This only became viable when they stole some genes out of the algae's nucleus that allowed for long-term survival of the plastids. Now we're going to talk about another means of horizontal gene transfer, and that involves transposable elements, also known as transposons. We're going to talk about some pioneering work that was done by somebody you may have heard of by the name of Barbara McClintock. This is her on the right. I had the great pleasure of meeting her one time, very briefly. She gave a talk at UVA when I was a graduate student. I think she was in her 80s when I met her. certainly late 70s. And she had more energy than the vast majority of any graduate student I ever knew. She was just a delight to be around, a high energy individual. Barbara McClintock described a system that involved what she referred to as a high frequency chromosome break at a location called a dissociation or DS site. The one she pioneered, the one she worked on, the DS site was inserted proximal to 3 genes: colorless, shrunken, and not shiny. Show you those in just a minute. In the case of the dissociation gene, she realized that a third gene, a third element was required that she referred to as the activator, or AC. So in the presence of the dissociation, as well as an activator, you would get chromosomal breakage. She found it impossible to map the activator sequences. Every time she tried to do recombination mapping, she got different values, which led her to suspect that the AC itself was mobile. It was moving around the genome. Here is the original one that she worked on. Here is the dissociation factor, DS. Here are the wild type alleles of the 3 genes. The most distal one, the one furthest away from the centromere, there it is so-called colorless, which is a bit of a misnomer because the kernel does have a color. It's just not the wild type color. Come on, arrow, right there. SH is shrunken, referring to the morphology of the kernel, and then not shiny, WX. If you mate this with a individual that is homozygous mutant for these 3 loci, and then you introduce a chromosome containing an activator sequence, in many cases you will see breakage and loss of the wild type alleles for these 3 genes, resulting in a portion of the kernel showing the recessive phenotypes. Colorless, which is actually yellow, which is the vast majority of the corn we eat today. Shrunken, the morphology is somewhat shrunken. It's not easy to tell in this photograph, and not shiny, which is also not easy to tell in this photograph. The purple represents wild type, pigmented, plump, and shiny phenotype. Yep, there's a dissociation factor. You can also have insertional mutants. The gene for pigmentation, the so-called colorless gene, is a recessive gene. You can have this dissociation factor inserted into the C gene, the colorless gene, causing a mutation, a loss of function. The other genes in the trio can remain functional if all it is an insertional mutagenesis, where the DS has inserted itself actually into the colorless gene, disrupting the coding sequence, causing what's referred to as an insertional mutation. If the other allele is C minus, then you get a colorless kernel of corn, yellow corn. In certain cells, you can have, in certain percentages, you can have the dissociation that departs and restores color. Now, this can only happen in the presence of activator, requires activator. Chromosomal breakage, as well as repair of the colorless gene, requires the presence of the AC element, the activator element. Both the AC itself, as well as the dissociation, the DS element, can insert into colorless and be remobilized by the presence of AC. AC inserts are always unstable because they're constantly moving around the genome.
Just a sec.
Sorry. I have no idea how it happens, but every once in a while, Siri thinks I'm talking to her. Here we have an example of the dissociation being inserted into the colorless gene, resulting in the vast majority of the kernel to be colorless, which is to say yellow, but in the presence of the activator locus. Can you guys see the arrow?
Yeah, we can.
Yes.
Because otherwise I'm wasting my time moving the arrow around if you can't see it. In the presence of the activator element, the dissociation will in some cases pop out, restoring functionality of the colorless gene. And that's how you can get these spots of colored purple in a yellow background, a colorless background. Just to illustrate, here's the wild type gene, what's called pigmented, which is purple in color. You have an insertion of the DS element into the colorless gene, disrupting the coding sequences, resulting in loss of the purple pigmentation. In the presence of the activator sequence, in certain situations, certain cells, the DS will be mobilized. It will relieve the gene, the colorless gene. It will ligate back, restoring functionality of the colorless gene, resulting in purple spots in an otherwise yellow background. In the case of AC insertion, it is autonomous. It doesn't need another factor, and an insertion by the AC into the colorless gene will automatically result in a mosaic of purple and yellow cells. She discovered what we now know as mobilized elements, transposable elements, in maize and corn in the 1940s. She was one of the pioneers in using corn in genetic studies. She got her PhD from Cornell. She got her bachelor's degree, master's degree, and PhD from Cornell University. She then went on to have a faculty position at the University of Missouri, and she worked at Cold Spring Harbor, which is a very prestigious investigative unit. The dissociation is non-autonomous. That is, it cannot function by itself. It requires the assistance of the activator, which is autonomous. That is to say, it's self-sufficient. It can mobilize itself. So today we would say the dissociation element is a non-functional transposable element, whereas the activator element is a functional transposable element. It is capable of mobilization on its own. She received the Nobel Prize in 1983 after transposable elements were basically rediscovered in Drosophila, or were discovered in Drosophila. People then went back and said, oh, this was what she was talking about. And so she got the Nobel Prize in 1983. She died about 9 years later. The good thing is that she had a very long life, and she lived long enough to reap the rewards of her early studies. So she was rewarded a Nobel Prize. If anybody has any interest in females in science in the 1940s, there's an excellent book. It's a very short book called A Feeling for the Organism. It details some of the trials and tribulations that she went through, as well as a lot of the accolades that she got for doing her work. I'm pretty sure before she got the Nobel Prize, she was a member of the National Academy, which is one of the highest honors any scientist can have bestowed upon them. So she was not looked upon in a negative light. It's just that the rest of the scientific world wasn't ready to understand what she was talking about with her mobile elements. As I said, fortunately, she lived long enough to reap the rewards and get the credit for what she had done. So the moral is, if you're going to be way ahead of your field, you better plan on living a long life if you want to get acknowledgments during your lifetime of your accomplishments. Gregor Mendel did not live long enough. He did not get any accolades during his lifetime. Yeah. What's going on? There we go. Okay, transposable elements can be broken down into two classes. Class 1 elements are RNA elements, which comprise about 42% of the human genome. RNA elements include things like the coronavirus, HIV, RNA tumor viruses, things like that. Elements whose genome is encoded in an RNA. So the genetic information in the mobile element, as it is being mobilized, is actually contained in a molecule of RNA. They're also called retroelements or retrotransposons. DNA is transcribed into RNA. Typically what happens is the transposable element somehow gets into the host cell. It takes its RNA, converts it into double-stranded DNA, and then that double-stranded DNA gets inserted into the genome, and then the virus, or the transposable element can be replicated through RNA polymerase transcription. Yep, RNA is converted into DNA and then it gets inserted into the human genome. A famous example in humans is referred to as ALU. It's a relatively short fragment, 2 to 300 base pairs. It's estimated that there's about 1 million copies within the human genome of this degenerate, they're all degenerate at this point, transposable element, class 1 transposable element. You can derive many copies from a single DNA insert because the mechanism relies upon transcription. Here shows you what happens. Typically, these transposable elements or viruses have a caspid. They have an envelope that surrounds a caspid, which contains the RNA genomes and an enzyme referred to as reverse transcriptase. Reverse transcriptase, you may recall, takes RNA and converts it into DNA. So once it gets inside the host cell, the capsid breaks down. Reverse transcriptase is used to synthesize a DNA copy of the RNA genome. It then synthesizes a second DNA copy. There's a specific enzyme that will degrade RNA in a DNA-RNA hybrid molecule. Now you have DNA that is capable of inserting itself into host genomes. You now have a proviral molecule inserted into the host genome. And this is what happens with HIV, happens with some of the herpes genes, various other genes, which is why they're so difficult to eliminate, is because they are now replicated by the same mechanism that we use to replicate our own DNA. The viral mRNA can be transcribed, producing literally thousands of copies. Gets packaged back in with the capsid and the envelope proteins, and now the virus can be infectious. Class 1 retrotransposons and related. They replicate the DNA, as I said, through RNA transcription, or they replicate the genome through RNA transcription. The retroviruses typically are flanked by what's referred to as long terminal repeats, LTRs. I'll show you some examples. There are 3 genes typically associated with these: GAG, POL, and INV. The GAG encoded protein is required for maturation of the RNA. The POL encodes for the reverse transcriptase, and ENV encodes for proteins surrounding the virus, that is, the envelope proteins. In the case of RNA tumor viruses, typically, in addition to these 3 genes, you find another gene, an oncogene, which is the gene that's actually causing the cancer to develop. So if you find a gene that's in addition to these 3 and it's causing tumors, it's a pretty safe bet that it's actually an oncogene, dominant gain of function causing cancer. If we get to the chapter on cancers, we will talk about different ways, different kinds of genes that result in cancer. These kinds of transposable elements are unique to eukaryotic organisms. Here's some examples just showing you the long terminal repeat. Here's the cassette of all 3 genes. Sometimes you have a slightly degenerate one. Taiwan in yeast, which is a fairly common transposable element in yeast, has the LTRs, but has lost the gene for the envelope. Same is true of Copia in Drosophila, which is a fairly common transposable element in Drosophila. In humans, there's an L1, which appears to have been derived from reverse transcriptase of the transcribed RNA from an original insert. You can tell that by the presence of a poly A tail. The poly A tail is not encoded in the original genome of the transposable element or the virus. It's estimated that, like I said, about 44% of the human genome is comprised of transposable elements. It's a huge component. We have long interspersed nuclear elements, anywhere from 1 to 5 kb in size. They are autonomous. They have all that's needed to be mobilized. The line is transcribed by RNA polymerase II. Sign, on the other hand, is actually transcribed by RNA polymerase III. It's a short interspersed nuclear element, 100 to 300 base pairs in length. It is non-autonomous. That is, it needs another factor to be able to mobilize itself. Lacks the reverse transcriptase. The ALU sequences that we talked about, roughly 200, a little over 200 base pairs, 200 to 300 base pairs, is one of these elements. So it's probably the most common element found in the human genome. It's named ALU because it's cut by the restriction enzyme ALU. There's a restriction site that's fairly highly conserved, and it comprises about 10% of our genome. It's also found in higher order apes, gorillas, chimpanzees, things like that, but not found in, at least to my knowledge, any other mammals. It's estimated that there are 20 times more transposable elements than genes that encode for proteins. So a huge proportion of our genome is actually transposable elements. Class 2 elements. If you have a class 1, you have to have a class 2. These are DNA elements. They do not have an RNA intermediate. The element itself or a copy has to move from the insertion site. Mobilization, as we talked about with Barbara McClintock's work, can result in a restoration of gene function in the case of insertional mutagenesis. The first elements that were discovered were actually this type of element, class 2 elements, DNA elements, the ones discovered by Barbara McClintock and Mays, as well as P elements that we're going to talk about somewhat extensively in just a minute or two. It's estimated that 0.2% of spontaneous mutations in humans are due to class 2 DNA transposable elements. Some examples include a couple of forms of hemophilia, breast cancer, other forms of cancer as well. P elements. P element story, I think, is a very fascinating one. Work on Drosophila melanogaster began shortly after the turn of the beginning of the 20th century, 1900s, and original researchers would literally go outside and capture existing fruit flies, what we now call fruit flies, Drosophila melanogaster. And if you look at wild type strains, you can see that their names indicate the location where they were originally captured. None of these contained P elements. So the ones that were originally isolated before the 1950s, there was no evidence of P elements being present. Today, the true wild type strains, not the ones we in the lab we call wild type, but the ones that congregate around your banana when you throw banana peel, when you throw it in the trash can, those almost assuredly all contain P elements. One way you can tell the difference between whether or not they are true wild type fruit flies or whether you've brought them back, if you're taking the lab, you've brought them back on your clothing, is how quickly they fly. You can almost catch the lab-bred strains in your hands. In fact, frequently you can. We have basically selected for slow-moving flies over the many generations. The wild-type flies, on the other hand, are much more nimble and virtually impossible to catch with your hands. So if you've got an energetic population of fruit flies, they probably came in from outside. If you've got slow-moving, yeah, you may have brought them home from the lab. The Russians, who did not have access to our fly stocks, went out and caught their own flies when they started doing genetics again. The story of the Soviet Union and genetics is a very interesting one. They rejected the idea of genetics for a long time because genetics implied some kind of a superior inheritance, and they wanted nothing to do with the Tsar, which was an inherited position. And so most of the good Russian geneticists when the Soviet Union came into being, were kicked out of the country. Cost them a huge amount of money in terms of their agriculture. Dobzhansky and people like that, particularly in population genetics, all came from Russia in this country. But in the 1970s, 1980s, they started doing genetic research again. As I say, the Russian scientists, what we now call Russian scientists, Soviet scientists, did not have access to our stock centers, so they had to go out and catch their own flies. Turns out that the flies that they caught in the wild all contained this transposable element known as a P element. The Western world, at the same time, were working with what is referred to as M cytotypes, and you'll see why they're named P and M in just a minute. But the key is the P cytotypes have P elements present, the M cytotypes do not. When you mate them in the proper direction, the result is something referred to as hybrid dysgenesis. When mom is the M cytotype, meaning the cytotype, there are no P elements in the genome, and dad is the P cytotype, hybrid dysgenesis occurs. This is why it's referred to the M cytotype, because mom has to be the M cytotype and dad has to be the P cytotype for mobilization to occur, for hybrid dysgenesis to happen. Hybrid dysgenesis is a pretty nasty thing, results in near complete sterility of offspring. You have a high rate of mutations, insertional mutations. You also have a high rate of chromosomal breakage. And it's probably chromosomal breakage that's the most deleterious of the whole thing. Chromosomal breakage and rejoining of chromosomes results in abnormal translocations, which really messes up segregation during meiosis, resulting in essentially sterile offspring. Here we show the cross, M cytotype females, P cytotype males. The F1 progeny are fine, but those F1 progeny cannot have any progeny of their own, or essentially none, because their gametes are just destroyed. The DNA inside their gametes are basically destroyed. If you do the reciprocal cross, where mom has the P cytotype, dad has the M cytotype, F1 flies are normal, no hybrid dysgenesis, complete fertility. This is just a table showing you that the only cross that results in hybrid dysgenesis, that this essentially sterile situation, is when pop is M type, P type, sorry, and mom is M type. The P type individuals contain an assortment of P elements, typically 200 to 300, many of which are dysfunctional. They're not autonomous, but non-autonomous, meaning they need the presence of a functional P element in order to become mobilized. There's a 31 base pair inverted repeat that flanks the element itself. It's a small element, roughly 3,000 base pairs in size. Transcribes an RNA with 1 small and 3 large ORFs, open reading frames. This encodes for an enzyme referred to as a transposase. Transposase is involved in the transposition of anything that has those 31 base pair inverted repeats flanking the DNA. Transposase recognizes the 31 base pair repeat, mobilizes any DNA that's located between the 31 base pair repeats, and causes reinsertion elsewhere in the genome, or can cause. Interestingly enough, the RNA is only properly processed to give a functional transposase in the germline. In the soma, there's an alternative splice that occurs. And let's say the P cytotype includes both autonomous and non-autonomous P elements, that is defective P elements. the defective P elements can produce a repressor molecule. That repressor molecule is stored in the P cytotype of unfertilized eggs. That is, the female, if she is P cytotype, she will store those repressor molecules inside the unfertilized egg. So that if that female's eggs are fertilized by a male who's also P cytotype, the P elements don't get mobilized because the repressor ensures that no mobilization occurs. In the case of the M cytotypes, they lack the repressor. So mom cannot store any of the repressors into the unfertilized egg. The introduction of P elements causes expression of the P elements. Its expression is not repressed. It immediately starts making duplicates and excising the P elements to be mobilized elsewhere in the genome. Transposase causes mobilization and insertion of P elements throughout the genome. It's essentially random insertion. This process causes insertional mutations, chromosomal breaks, and results in essentially sterility excuse me, in the germline. This is the intron. This intron, which is present in the somatic cells, contains a stop codon terminating translation of the enzyme transposase prematurely, resulting in a non-functional transposase in the somatic cells. But in the germline, it gets an alternative splicing, removes this intron, and allows for production of a functional transposase. The organism, from an evolutionary perspective, they don't want to damage the somatic cells. Where they want the P elements to be mobilized is in the germline, and that's where the P elements are going to get perpetuated. They don't want to kill their host, but they do want to perpetuate their own genome. This is the P element. Here we have the introns inserted inside the transposase gene. The best we can hypothesize, speculate, and compelling evidence for is that the P elements appear to have been introduced into wild populations, that is, horizontal gene transfer, after the US labs had collected all their wild type strains. Early on, something somewhat similar to hybrid dysgenesis in that matings result in sterile offspring. Fly labs would exchange stocks only to find out they couldn't mate them with each other. Turns out that they discovered a species of Drosophila simulans, that is, in the adult, morphologically indistinguishable from Drosophila melanogaster. You can only tell the difference during the larval stage, and it has to do with the number of teeth, if you will, that the larvae uses to feed itself. That's the only way, the only morphology to distinguish the two. The Russians basically discovered hybrid dysgenesis. Used to go to fly meetings all the time. A lot of fun, pretty grueling though. The meetings would typically start around 9 o'clock in the morning, and they typically would not end until 8 or 9 o'clock at night, with 15-minute talks interspersed with 1-hour talks throughout the day. There was tremendous amount of information packed inside those. By the way, the proper pronunciation of the Drosophila is Drosophila. It is not Drosophila. Occasionally you'll see movies where they mispronounce the name and call them Drosophila. I was at meetings one time in Chicago where they had a sign up. I was sitting at a bar having a Coke or something, and there was these two well-dressed ladies sitting a seat away from me at the bar. And one of them, they had signs up for the meetings. One of them turned to the other one and said, "What the hell is a Drosophila?" Talked about the International Drosophila meetings. And the other one looked at her and said, "Oh, shit." They're scientists. Let's get out of here. We're not going to make any business tonight. That's supposed to be funny. They were obviously prostitutes. They left. But it's Drosophila, not Drosophila. P element appears to have been inherited or acquired from a distinct species of insects. There are two types of mites that are the bane of existence of Drosophila melanogaster research labs. One is a white mite, which is about the size of the Drosophila egg. It competes with Drosophila adults for the food. And if you have a weak stalk, the white mites can overcrowd the stalk, and it makes it difficult to maintain a weak stalk. The others are more insidious, and they're a small red mite. And those are much smaller than the white ones, and they actually feed upon the eggs of Drosophila melanogaster. So the white ones feed on the food, the red ones feed on Drosophila itself. There were insects that the mites fed on, not just melanogaster, but some other ones, one of which is believed to have been the source of the original P elements. It's postulated that through horizontal transmission, the P elements were introduced into melanogaster. The story goes that what they thought happened was the red mite was feeding upon this other insect in which P elements were originally found. Their feeding was interrupted by some means or another. They migrated on and found melanogaster eggs and completed their meal on the melanogaster eggs. In the process, transferred P elements in their saliva into the egg. They didn't consume the whole egg. That egg grew up, produced an adult, and the introduction of P elements into the genome was accomplished. Within a few decades, essentially all true wild-type strains now contain this transposable element. I think from an evolutionary point of view and the rapidity with which these P elements spread throughout the population is pretty remarkable. Not only is it an interesting story from an evolutionary point of view, but also because of insertional mutations, P elements can be used to tag different genes. And a lot of early cloning of genes relied upon mutations that were caused by insertional mutation by the P elements that then provided a tag, if you will, for the mutated DNA. The P element will insert inside a gene, much as the dissociation factor inserted into colorless in corn. It disrupts the gene's function. But now, because the P element resides inside the gene you're interested in, it acts as a tag. You can make a library of such a mutant that you know the mutation is caused by the insertion of the P element. You can use P element DNA, clone P element DNA, as a probe to your library to identify clones that contain the P element, but will also contain genomic DNA encoding for the gene that you're interested in. Can be used to clone inserted gene, especially if you're using a slightly larger vector. Plasmids typically are good up to about 6,000 base pairs. Bacteriophage, Lambda in particular, are good for up to about 20,000 base pairs. So if your library is in a phage, you get a 20,000 base pair clone, which will almost always include DNA for the gene that you're interested in. One way to confirm that the mutation is caused by insertional mutagenesis, the insertion of the P element, is by remobilizing the gene to restore function, much as we saw, again, in maize, where you restored the functionality of the colorless gene. So you had a yellow kernel with a yellow background with spots of purple, where the gene's function had been restored. You can do the same thing in melanogaster with P elements. That's considered conclusive proof that the mutation was actually caused by insertional mutagenesis. You can use the 31 base pair repeat to conduct gene therapy in flies, and we've been doing gene therapy in flies since the 1980s. You can insert any DNA between that 31 base pair repeat, and it will get inserted into the genome. We're getting close to the end. We weren't dropped, so it looks like the attempt at circumventing just the use of WVU site is. Are you all still there? Hello?
Yes.
Okay. Feel free to ask questions anytime during the talk. The insertion is essentially a random insertion. Once inserted, you can remobilize it if it's an insertional mutation and you want to confirm that it actually inserted into the gene that you're interested in, and it's not simply a spontaneous mutation occurred simultaneously with mobilization of the P elements. You can reintroduce transposase, get remobilization, and restore function in at least 1 to 10% of progeny. This is just an illustration. Here's the transposase gene. Here's the 31 base pair repeat. This is a functional P element where it acts autonomously, but you can replace this transposase DNA with any DNA that you have an interest in. And this is how the collection of GAL4 transcription factors were collected. You had GAL4 with a minimal promoter, no enhancer elements, as well as typically GFP, which allows you to monitor the insertion site. You mobilize this thing and get it inserted all over the genome, and its expression pattern is going to be determined by the site in the genome into which it inserts, under whose enhancers its expression is determined. And that's how we have this huge collection of GAL4 expressing lines in Drosophila. This is how gene therapy is done. The posterior end, which is where the gametes are going to be formed, you take a needle, glass needle, you have your clone DNA with the 31 base pair repeat flanking the gene that you're interested in. In this case, it's a gene RY, rosy, which is involved in eye color pigmentation. At the same time, you will inject another bacteria plasmid that contains the genes for the transposase. So you have a source of the transposase as well as what you want in the transgenic construct. You inject both of them simultaneously into the region where the gametes are going to form. Hopefully, if you're lucky, some of these gametes will have taken up your transgenic construct into their genome. You then take that fly and you mate it with a homozygous mutant, or Rosie in this case, and you look for flies where Rosie has been rescued, indicating that the transposable element has inserted itself somewhere in the genome with the wild type copy of the Rosie gene. And you can do that with any gene in the genome. We used to do that here in, not here, but in Brooks Hall, when the biology was Brooks Hall. Making these transformations is not trivial. They're only about 10% successful. Now there are companies that will do it for like $100 each transformed line, which, believe me, is a real bargain compared to the hours you spend trying to get it done yourself. Okay, at this point, we're going to stop for today. Does anyone have any questions? Okay, I'm going to review the notes and decide whether or not we're going to do HIV next. Let's do HIV next. So I will be sending you the PDF for HIV. We will talk about HIV. A lot of it's going to be historical, but we're also going to talk a little bit about some of the issues with HIV. I should remind you that while we're all optimistic that we're going to come up with a vaccine against the coronavirus, don't be too optimistic. We have been for at least 30 years trying to come up with a vaccine against HIV. Literally billions of dollars have been spent trying to come up with an effective vaccine, and we don't have one. Another coronavirus, the common cold, a lot of effort, not nearly as much as with HIV, but a lot of effort has been spent trying to come up with a vaccine for that, and we've been unable to do so. The immune response is an illustration of our ignorance regarding integration of the different components. We know all the players in the immune response. What we don't know is how they interact with each other to result in traits and characteristics that you would not predict looking at the minimalist, looking at individual components of the system. We don't know how they integrate to have produced a functional vaccine. So, bit of a bummer. Saw a study where 70% of Americans say they will never attend a sports event in person until after an effective vaccine is developed for the coronavirus. I think it could very well signal the end of sports as we know it in this country. If there's no questions, I will let you go. As I said, I will be finishing the grading. For those of you, there's 10 or so left that I need to finish grading. I will be sending them out individually with comments. If you have any questions, I've sent you all the answer sheet already. Look at the answers, look at my comments. If you have any questions, feel free to send me an email. I will address them. I will be sending you the PDFs for the HIV, and I will hopefully be getting this thing loaded up to YouTube, at which time I will be notifying everyone. And I know it's going to be somewhat embarrassing because I forget that the sound is on from the moment I establish a link. So you may end up hearing me talk to my wife occasionally. Anyway, everyone stay safe, stay healthy, and I'll talk to you on Thursday. That's it, folks.